Hey guys, welcome to Brainiac Pharma. In today's class, we shall be looking at G protein coupled receptors. These are popularly called as GPCRs, and well, there are four popular subtypes of these G protein coupled receptors, which are GS, GQ, GI, and G12. Of these four, we shall be looking at GS and GQ primarily. Now, what are these G protein coupled receptors? Now, these are the receptor subtypes onto which a endogenous neurotransmitter would act as an agonist and show an intrinsic activity. Before getting into the subtypes and their functions and the internal mechanism through which they act, we should be first trying to understand the basics of how a cell membrane is arranged and how these G protein coupled receptors are arranged across the cell membrane. Well, we all know a soap molecule. A soap molecule is generally represented with a polar head and a non-polar tail. Okay, so this region is polar, okay, where the salt formation occurs. This is the non-polar part. So this is the salt of the fatty acid and this is the hydrocarbon chain of the fatty acid. So this is a soap molecule. When we talk about cell membrane, okay, we say that cell membrane is made up of a phospholipid bilayer, and this phospholipid bilayer is represented with a polar head and two non-polar tails. Okay. So before looking at the arrangement of a cell membrane, we'll have to understand why it is arranged in that manner. So what exactly and how exactly a cell membrane is arranged. Consider two scenarios. The first instance, a container of water. The second instance, a container of oil. Okay. So this container has oil. Sorry, it has water. This container has oil. Now, if I am going to add the soap molecules into this container of water, the orientation of these soap molecules will be such that all the polar heads will have an accommodativeness. It will have an affinity, okay, to the polar water. So they go into the solution. However, there is an incompatibility with the polar solution of the solvent and the non-polar tail of the soap molecule. It is because of which the non-polar tail in these soap molecules will start orienting outside in the air. Why? Because there is an incompatibility. So this would be a good representation of how a soap molecule, how a bunch of molecules, when they are subjected to water in a container, would arrange themselves, accommodate themselves. The same soap molecules, if I am going to add in a container of oil, what happens is, because of incompatibility of oil and the polar heads, because oil is non-polar, the polar heads would orient themselves towards the outside and the non-polar tails would come into the solvent. You see? Right? Now, this is how you see the total difference. This is how the same soap molecule would orient itself when it is subjected to a container of oil. The same soap molecule when it is subjected to a container of water. Now, what would happen if I add more amount of soap molecule in this container of water? The surface of this container where the soap molecules interact with water, it gets saturated. Right? Once it gets saturated, there is no scope for any space to be left here. All the soap molecules would occupy the surface of this water on the upper side. Now, if I further continue to add the soap molecules when they are already saturated, what happens? Well, this is what happens. So please look here. So the container has water. So we have seen that there is saturation of these soap molecules. Okay, we are considering the scenario where there is no space, it is completely saturated. We further add the soap molecules. When we keep adding the soap molecules, what happens? There is no space for them to get accommodated. So all these soap molecules will now move into the solution. How do they move into the solution? They move, upon saturation, they move in this manner that all the polar heads will come together, leaving, see, all these polar heads have come together, forming a kind of a spear. You see, outside it is all polar, and because outside is polar, so this is the case in which you are seeing water, right? So outside is polar, these are polar heads, there is a compatibility. Now what happens to the non-polar tails? The non-polar tails will come into the solution or will come into this sphere such that all the non-polar tails which have affinity, they come together, pushing out all the water in this space. So this particular space inside the sphere does not have water because there is an incompatibility of these non-polar tails with that of the water, which is polar, and all this water is pushed out and they interact with the surface of the polar heads. Such a spherical formation of a soap molecule inside water, okay, or oil, which we shall be seeing, is called as a missile. You see this? This is the first instance in which we have added a polar soap molecule into a polar solvent, where first all the surface of the solvent container has been saturated with the heads of the soap molecule. We further keep adding, when we further keep adding, the saturated soap molecules which were on the surface now go into the solution and orient themselves in such a way that all the polar heads come together and they have an interaction with the polar solvent, and the non-polar tails, they come together pushing all the water molecules out and form a sphere. These spherical structures are called as missils. Now, what happens if we keep adding the soap molecule in a container of oil? In the same way, we have seen, considering this to be a container of oil, right? So this is oil which is non-polar in nature. In this, we have added the soap molecules. When we have continuously added these soap molecules, the surface is saturated. The surface is saturated. Now, in spite of the saturation, I continue adding these soap molecules. When I keep adding these soap molecules, what would happen? All these non-polar tails would orient themselves outside You see this? The outside is oil. The oil layer is, see this, this is oil. There is a compatibility with the non-polar tails and the oil which is present here, and there is an incompatibility of the polar heads and the oil. In order to balance this compatibility issues, they once again form a sphere. But in this case, the sphere is formed in this way that all the heads, they go into the solution, where this sphere does not have any oil left over, but all of this oil is pushed out. You see, this is again a sphere. It is just the opposite to that of what happens when more than saturated point of soap molecules are added in a container of water. When they are added in a container of water, they orient themselves in this manner. See, such that the tails are inside and they push all the water out. But when the outside, when the solvent is full of oil, all the non-polar lipophilic tails, they orient outside and the polar heads come inside, where the space here does not contain any oil left. Understand? This is a scenario. So this is a micelle formation in water. This is also called as a micelle, and this is its formation in oil. Fine. So we have seen, because of the compatibility issues, because the external outside surface contains non-polar region, all the non-polar tails are oriented outside and the polar heads have come inside. In case of water, the outside region is completely polar, so the polar heads are oriented outside and the non-polar tails have gone inside, where this space does not have any water left in it. Now what happens in a cell membrane? Right? Now what exactly happens in a cell membrane? See this? So this is a cell membrane. Okay. This is a cell. Okay. Cell membrane. We say that cell membrane constitutes a phospholipid bilayer. See this? Constitutes a phospholipid bilayer. What exactly is this phospholipid bilayer? You should remember that, considering this to be a phospholipid bilayer, outside, okay, let me mark it with a better color. Outside there is, this is nothing but your blood, right, which is the extracellular fluid, and inside there is cytoplasm, okay, cytosol, right, so this, or the organelles, okay, inside there is cytosol. Outside there is blood, inside there is cytosol, and covering the cell is the cell membrane, which constitutes phospholipid bilayer. Now how exactly is this phospholipid bilayer made? See, we have seen the problem. So when we have seen a soap molecule, so this is just like a soap molecule. Okay, there are two non-polar tails and a polar head. Say, if you consider the phospholipid bilayer, or if you consider the cell as such, if you take the cell, outside of the cell is polar, inside of the cell is again polar. Now we have one layer constituting a non-polar tails, the other layer containing polar head. So what we need to accommodate this, or the body needs to accommodate this phospholipid bilayer, with phospholipid, in such a way that it would meet, or it would have no compatibility issues with the polar extracellular fluid, and it does not have any compatibility issue with the polar intracellular fluid. In that case, the arrangement changes. Now how is this arrangement done? You see this, We have already seen the micellar formation. Similar to the micellar formation, the phospholipid within the body, consider this as a part of a cell membrane. So this is a cell membrane from this region to this region. So this region I am representing it here. Okay. See, these are the polar heads with two non-polar tails. Right. Now the inside of the cell is polar, and these are non-polar tails which cannot accommodate or which cannot be compatible with the polar region. So what happens? Body does something which is a kind of a genius. These non-polar tails once again interact with another non-polar tail. You see this? Because these are non-polar and the non-polar tails of the next phospholipid is also non-polar, this non-polar come together, and the polar heads of this secondary phospholipid is arranged in this way. You see this? Now, this is this region of the cell membrane. The outside contains a polar blood. Now, inside is the polar cytosol or cytoplasm. See this? This phospholipid bilayer surrounds the cell, right? This is the constituent of the cell, where this phospholipid bilayer, one after the other, it is arranged in such a spherical way that it is based on the principle of the micellar formation. But you see this, the micelle is so grandly arranged, it's a kind of a genius work by the body, such that the cytosol, which is polar region inside, is accommodated with this polar head because the entire surface is polar. The outside blood is also polar, which once again has no issues with the polar heads outside. That is why the phospholipid, which is part of the cell membrane, is called as a phospholipid bilayer. Fine. So all the cell membrane constitutes phospholipid bilayer. So we have been looking into these phospholipids, right? We say that this is a phospholipid bilayer and this is how a phospholipid is arranged. But what exactly is the chemical constitution of this phospholipid bilayer? I hope this is clear. Well, we'll have to see now how this phospholipid bilayer is arranged. So we've been talking about phospholipid bilayer. What is its constitution? All these phospholipids are glycerol derivatives. Basically, they are triesters of glycerol. So the backbone which is present is a glycerol backbone. So it's a CH2OH. Okay, so this is basically glycerol. Now, alcohol reacts with acid to form ester. So, glycerol, within the body, there are endogenous fatty acids, right? So, there's a long chain fatty acid which combines with glycerol, right? CH3, CH2N times, okay, CH2, COOH, right? This is a long chain fatty acid which combines with this glycerol to form a monoester of this okay. Let me erase this. So this is called as monoacyl glycerol. Fine. endogenously, if we consider this combining with another fatty acid, right? Now what happens? There is another ester that is formed within the same glycerol moiety. So this is glycerol monoester. Just to differentiate, I am using red ink. This is N times. CH3. Right, so this is called as DAG, which is diacyl glycerol. And similarly, when there is an esterification at the third alcohol, we call it as triacyl glycerol. But most times within the body, what we find is the esterification that happens, it happens with derivatives of phosphoric acid. When phosphoric acid derivatives are found in the body, we can assume that diacyl glycerol combines to form an ester here with phosphoric acids. Ultimately, this would lead to the formation of phosphoric acid derivatives, right? One of such phosphoric acid derivatives which is most popularly found within the body is this. So it is CH2O, CHO, CH2O. See this PO minus. This is the phosphoric acid derivative. This is C double bond O, CH2, CH2N times CH3. Similarly, C double bond O, CH2, CH2N times CH3. See this. This is the phosphoric acid derivative. Okay. When diacyl glycerol, okay, combines and forms an ester with phosphoric acid, the whole molecule that we get, which is very commonly found within the body, is called as phosphatidic acid. So this is basically phosphatidic acid, and the phospholipid bilayer is a derivative of this phosphatidic acid. You see, this phospholipid representation was given by Nicholson and Singer. Nicholson and Singer had given this representation only to accommodate and show that these phospholipids are basically phosphatidic acid derivatives. So this side, because of the presence of ionic ends, this phospholipid is simply represented with a polar head. Because of this, there is polarity, and two non-polar tails. See this? This is the ultimate representation. So we have been seeing from the beginning that a phospholipid has a polar head and two non-polar tails. Because these are derivatives of phosphatidic acid, okay, as these are derivatives of phosphatidic acid, right, it has a polar head and two non-polar tails, and these are phospholipids because these are lipophilic in nature, so these are called as phospholipids, right. Endogenously, there are within the body different types of phospholipids, and this phosphatidic acid will further form esters with different alcohols within the body. So some examples, lecithin is an example, right? Cephalin is an example. right? So it's like CH3. I'm just representing this in the opposite way. N times CH2, C double bond O, CH2, CH, CH2, O, C double bond O, CH2, CH2, N times CH3, O, PO minus. So this is phosphatidic acid. Now this phosphatidic acid within the body, it forms esters. So this is an acid, right? So in phosphoric acid there are three areas through which it can form ester bonds. So through one of the acidic ends, it combines with endogenous alcohols, many times endogenous amino alcohols, to form esters. Okay? If the endogenous amino alcohol is a choline, that is CH2, CH2, and CH3, CH3, CH3. See this? This is choline. Okay? Okay, ethanolamine. Okay, this is trimethyl ethanolamine. This is choline. Just like we know that from choline comes acetylcholine. Okay, the neurotransmitter for the cholinergic system. So, if this forms an ester, see this is an ester. When it forms an ester, phosphoric ester with choline, you get phosphatidyl choline, because this was earlier phosphatidic acid, and now once it has formed an ester with choline, you get phosphatidyl. You get phosphatidyl choline, which is also called as lecithin, right? Okay, phosphatidyl choline, which is also called as lecithin. Okay. If the ester is formed with inositol, okay, okay, so this is inositol. Inositol is nothing but there is 6 OH groups attached to all the 6 carbons of cyclohexane. So this is inositol. So if one of the alcohols form an ester, so this is simply called as phosphatidyl inositol. See, this is called as phosphatidyl inositol. So transferases are a set of enzymes that tend to transfer an essential group in the process of activating. Phosphorylation is generally is part of these transferases belonging to EC2 classification. As per the enzyme classification numbering, it belongs to the subclass of EC2. It belongs to the class of EC2. In that, there is a subclass called as kinases. So all these kinases tend to phosphorylate an alcohol, okay, to form it as to make it a phosphate derivative. Okay, within the body, phosphatidyl inositol is found to be existing. In phosphorylated state. Further, there is further activation. Okay, this is PO4. Okay, phosphate is basically PO4 3-. So this is further phosphorylation of the phosphatidyl inositol, right? So the phosphorylation of phosphatidyl inositol would yield in the formation of phosphorylated phosphatidyl inositol. So this gives us phosphatidyl inositol 4,5 bisphosphate, right? So this is 4,5 bisphosphate. See, 2, 3, 4, 5. 4,5 bisphosphate. Phosphatidyl inositol 4,5 bisphosphate, popularly called as PIP2. It is an endogenous ester of phosphatidic acid which is commonly found in the cell membrane. Just like lecithin is commonly found in cell membrane. If it is a simple ethanolamine, it is called as cephalin. In that way, we find all these constituents which are basically part of our cell membrane, right? So this is nothing but your polar head with two non-polar tail, and if it is phosphate, if it is PIP2 which is part of the cell membrane formation, then still this justifies the polar head and two non-polar tail representation, which we have been seeing right since the beginning of this particular class, which is the Singer and Nicholson's representation of a phospholipid. And these phospholipids, these lipid bilayer come together, okay, and the polar surfaces orient either outside the cell or inside the cell, right? And these are the different types of phospholipids which are found in the cell membrane. Now, coming back to understanding how the G protein coupled receptor exists and how does it work. Now we have understood how a cell membrane is. So I am simply representing one small area of the cell membrane, which is a phospholipid bilayer, right? Right, this is a phospholipid bilayer. Now, first of all, as we have seen that G protein coupled receptors have GS, GQ, which we are going to deal mainly. First, we will see what is a G protein coupled receptor, and we begin with GS pathway. A G protein coupled receptor is a 7-pass transmembranal receptor. Fine. It is a 7-pass transmembranal receptor which is coupled to a protein called as G protein. Okay. When I say 7-pass transmembrane, see this, when I say transmembrane, it means that it traverses the phospholipid bilayer from the intracellular region and the extracellular region. So, you see, this is blood flowing here. Let us consider this to be the extracellular region, and this is the intracellular. Okay. Okay, so there is blood here, right? And then there is cytoplasm here. Okay. Now, what exactly is this G protein coupled receptor? So this G protein coupled receptor, let me use the red marker for convenience and ease of understanding. There is a domain on the extracellular region, okay, which is simply called as the amino terminal, okay, because it is all receptors are proteinaceous in nature. It has a carboxylate terminal and an amino terminal. In case of G protein coupled receptors, the amino terminal is towards the extracellular region and the carboxylate terminal is towards the intracellular region. Now, this is the the neurotransmitter binding domain. Okay, it is here that the neurotransmitter, which is now present in the blood and released from the neurons, okay, of the autonomic nervous system, they travel through the blood, right, and they find a particular receptor towards which they have the affinity. Once this particular molecule of the neurotransmitter has an affinity, it goes and binds to that or interacts with that particular receptor, right? That is what we generally tend to call it as the drug receptor interaction. Before that, see this is called as a 7-pass transmembrane receptor. So when I say 7-pass, it passes through this phospholipid bilayer 7 times. Okay, 1 time, 2 times, 3 times, 4 times, 5 times, 1, 2, 3, 4, 5, 6 times and 7 times. So inside is the carboxylate terminal. So outside is the amino terminal, inside is the carboxylate terminal. This is a G protein coupled receptor where the receptor traverses the cell membrane 7 times. That is why it is called as 7-pass transmembrane receptor, where towards the intracellular region it has a gamma subunit followed by a slightly bigger beta subunit followed by a much bigger alpha subunit, and these 3 subunits are part of G protein. Okay, let me erase this. Inside is zero amino terminal. Okay, just to differentiate that, right? So this is the receptor which has a extracellular amino terminal, intracellular carboxylic terminal. 7 times it traverses the phospholipid bilayer, and towards the intracellular region there is a G protein. The G protein consists of 3 subunits: gamma subunit, right, beta subunit and alpha subunit. The gamma subunit is smallest in size, followed by beta subunit, which is slightly larger, followed by alpha subunit, which is larger than the other 2. Right. So this is what is the basic structural features of a G protein coupled receptor. Now, this alpha subunit of a GPCR, which is G protein coupled receptor, has a GDP moiety attached to it in the intracellular segment. So this completes the structure. The difference in sizes in my writing represent the fact that the alpha subunit, see, this is with GDP. Alpha subunit is larger than the beta, which is larger than the gamma subunit. Now, this alpha subunit is associated with GDP. So GDP is guanosine diphosphate. Right. So this is the setup of a G protein coupled receptor. It's a 7-pass transmembrane receptor which traverses the cell membrane 7 times with an amino terminal on the extracellular region and then a carboxylic terminal on the intracellular region. This in the intracellular region, a G protein is attached to the receptor, which has 3 subunits: alpha subunit, beta subunit and the gamma subunit, and the alpha subunit is attached to a GDP moiety. Now, when an endogenous agonist, okay, now let us consider the first scenario. Let us consider this to be a GS subtype of receptor, okay, GS. The difference comes in the type of the alpha subunit. That is why many times this means this can also be represented as G alpha S subtype of receptor, GS subtype or G alpha S, because the alpha subunit is what is showing the difference in the activity. Now, the moment I say this is a GS subtype of receptor, you should remember that the cell membrane along with the GPCR, it constitutes an enzyme which is called as adenylyl cyclase. Now remember, the moment I say that this is a GS subtype of receptor, it means the GS subtype is always associated with a adenylyl, with an enzyme called as adenylyl cyclase or adenylate cyclase. It's just the same. The names are, there's a small change in the difference, adenylyl or adenylate. Fine, adenylate cyclase or adenylyl cyclase. Now, so this is a G alpha S subtype of GPCR, right? The moment an endogenous agonist, right, when it interacts with the receptor, there is a change that takes place within the receptor inside the cytosol such that ultimately a endogenous activity is shown. Okay, when a moiety has an affinity and shows an intrinsic endogenous activity, it is called as an agonist. If it simply comes and sits, preventing every other molecule which has affinity here to bind, preventing that, if it simply comes and sits without showing any intrinsic activity, it is called as an antagonist. An antagonist has affinity to the receptor without any intrinsic activity. So we are looking at the case of an agonist. So a GS subtype, let us consider to be a beta-1 receptor in this instance. So the noradrenaline moiety, okay, the noradrenaline moiety, say it comes and binds to the receptor, it has an interaction. The moment a receptor comes and sits in the receptor binding domain, okay, of the G protein coupled receptor, which is of the GS subtype or G alpha S subtype, the alpha S GDP immediately gets phosphorylated through series of mechanisms within the body such that the alpha S GDP, that is guanosine diphosphate, now gets converted into alpha S GTP, which is guanosine triphosphate. See, earlier it was alpha GDP. The moment an endogenous agonist comes and binds to the receptor site, the alpha GDP gets converted into alpha GTP. The moment GDP gets converted into GTP, this alpha unit which is attached to the beta gamma unit no longer will be able to hold itself. So it detaches and comes out into the cytosol as alpha S GTP. You see, the beta gamma subunit stays there. Okay, the beta gamma subunit, it remains, it stays within the cell membrane and the receptor. So this alpha S GTP comes into the cytosol. Agonist sits onto the receptor binding domain. Alpha S GDP gets converted into alpha S GTP. The alpha S GTP detaches itself from the beta gamma subunit. So this is simply referred to as beta gamma subunit. This is the alpha S GTP subunit. It detaches and comes into the cytosol. The moment alpha S GTP comes into the cytosol, this alpha S GTP, so this is adenylyl cyclase, right? This alpha S GTP activates adenylyl cyclase. So adenylyl cyclase all this while was in an inactive state. The moment alpha S GDP gets converted into alpha S GTP and detaches from the beta gamma subunit of the G protein, this alpha S GTP, which is inside the cytosol, activates the inactive adenylyl cyclase, which now becomes active. The moment adenylyl cyclase is activated, right? So you can simply write it as inactive, okay, adenylate or adenylyl cyclase to active adenylate cyclase by alpha S GTP. See, inactive adenylyl cyclase now becomes active. The moment adenylyl cyclase becomes active, right? It converts ATP, adenosine triphosphate, to cyclic adenosine monophosphate. See this? So ATP is present within the cell. Okay, this ATP so this is OH, CH2O phosphorylations. Okay, this is simply ATP. So the adenosine triphosphate, ATP, gets converted into cyclic adenosine monophosphate. The moment cyclic adenosine monophosphate is formed, there is within the cell a protein kinase A. An inactive protein kinase A gets activated simply called as pKa. An inactive protein kinase A, which is responsible for the intrinsic activity within the body, gets activated by this cyclic AMP, and this inactive becomes active. This active pKa will show intrinsic activity or endogenous activity. You see, there is agonist binding to the receptor site. Alpha as GDP gets converted into alpha as GTP. The moment alpha as GTP is formed, it detaches itself from the beta gamma subunit of the G protein. When it detaches itself from the beta gamma subunit of the G protein, it activates the inactive adenylyl cyclase to active adenylyl cyclase. This active adenylyl cyclase converts adenosine triphosphate to cyclic adenosine monophosphate. This cyclic adenosine monophosphate activates an inactive protein kinase A which is present within the cell to active pKa. This active protein kinase A is responsible for the intrinsic activity that is shown in the GS subtype of G protein coupled receptors. Now, as long as there is cyclic adenosine monophosphate generated within the body, within the cell, there is continuous activation of pKa, that is the inactive becomes active, and this active pKa will show intrinsic activity. So whenever there is a generation of CAMP, there is intrinsic activity that is shown. What is responsible for the CAMP generation? Active adenylyl cyclase. What is responsible for adenylyl cyclase becoming active? Alpha S GTP. How does alpha S GTP form? Whenever there is an agonist which binds to the receptor of GS subtype of G protein coupled receptors. Now, as long as CAMP is present, you have intrinsic activity, but the body does not tend to continuously show this intrinsic activity. There are many ways in which it shows the activity, and it is also important to stop the activity because excess activity is not required. So consider GS subtype is present in the beta 1 receptors of the heart. If there is an increase in heart rate, body will not continuously keep on having a heart rate which is more than normal, which is supranormal. No, it would increase the heart rate whenever it is required and suppress the heart rate in trying to maintain the homeostasis. Of course, whenever there is adrenergic system acting, there is also the parasympathetic or the cholinergic system acting in tandem with this in order to maintain the homeostasis. Even within the adrenergic system, whenever the body is acting, whenever the cell is showing an activity, it shows the activity, and after a certain period of time, it has to stop the activity. And how does this, the normal way in which this activity gets stopped? It is either by inhibition of the CAMP by the body itself. The moment CAMP is inhibited, there is no longer any intrinsic activity that is formed. So this is one way in which the CAMP is inhibited in order to prevent the activity that is shown within the body, and the other way is, of course, there is the detachment of the endogenous agonist. The moment agonist detaches itself from the receptor site, the whole of this series of events which happens are reversed. Again they get back into normalcy, because this one cycle of event takes place. For the next cycle to happen, there is no longer the stimulus. The stimulus is removed, and then the receptor gets back itself into the parent position, where the alpha GTP becomes alpha GDP, and similarly active becomes inactive. CAMP is once again broken down, and further ATPs are generated within the cell, and the active pKa gets back to its state of inactive pKa. But in this conversion of ATP to CAMP and the inhibition of CAMP, we shall just look at this in a little more detail. See this, there is this is our adenosine triphosphate. This adenosine triphosphate, as we have seen, ATP, in the presence of active adenylyl or adenylyl cyclase, gets this ATP converted to cyclic AMP. So cyclic adenosine monophosphate is what is formed. See this. Right. See this. This is 1, 2 CH2 O. See this. This is 1 prime, 2 prime, 3 prime, 4 prime. The ester which is formed is between the 3 prime and 5 prime position of this adenine moiety. Okay, of this nucleotide, okay, this adenosine, which was adenosine triphosphate, because of the presence of the influence of adenylyl cyclase, there is a cyclization that takes place. Okay, this is the 1 prime, this is the 2 prime, 3 prime, 4 prime, and this is the 5 prime position. There is a cyclization that takes place, okay, between this phosphate moiety and this OH to form a 3-5- ester. Okay, so this is what is called as cyclic adenosine monophosphate. See this? This is called as CAMP. Okay, so this is a 3-5- ester. So there is an ester bond with this phosphate moiety or this phosphoric acid moiety. There is an ester through this alcohol at the 3rd position and the alcohol at the 5th position. Understand? So this is a diester which is formed here, which is cyclic adenosine monophosphate. Right? So this cyclic adenosine monophosphate is formed from adenylyl cyclase. It is formed from ATP in the presence of adenylyl cyclase, which is more specifically called as 3-5- cyclic adenosine monophosphate. Now, we have seen that this cyclic AMP will activate the protein kinase A, right? The inactive protein kinase A to the active protein kinase A, and the active protein kinase A will show the intrinsic activity. But this activity needs to be controlled, and the control happens when the cyclic AMP is inhibited by an enzyme, endogenous enzyme called as phosphodiesterase. You see, this is simply called as PDE. Okay, depending upon where this phosphodiesterase is present, it is classified into several subtypes: PDE5, PDE3, so on and so forth. Okay, so when it comes to heart, when we are talking about beta-1 receptors, then this particular subtype is PDE3, phosphodiesterase 3 subtype of enzyme. So this enzyme, PDE enzyme, you see, we have already seen that this is a 3-5- diester. So there is this diester. So diesterase enzyme will break this diester to give a monoester. So it breaks at the 3' position in such a way that it leads to the formation of cyclic adenosine monophosphate, will lead to the formation of the OH is restored and there is CH2O PW. So this is a monophosphate that occurs at the 5th position, simply called as 5-adenosine monophosphate. So CAMP leads to the formation of 5-adenosine monophosphate in the presence of PDE. When we are talking about beta-1 receptor, this is essentially phosphodiesterase 3 subtype. Understand? So this is how the cyclic AMP gets converted into 5-AMP. The moment CAMP is converted into 5-AMP, there is no longer CAMP that is available in order to activate the protein kinase A, and then the cell gets back to its original or the parent position in order to be once again activated by an endogenous agonist. So this is the full cycle of a G-alpha S subtype of receptor. Because of the presence of adenylyl cyclase, this is also called as the adenylyl cyclase pathway, right? Or because of the presence of CAMP, this is also called as the CAMP pathway. We should also remember that within the cell, this particular CAMP will not only activate the protein kinase A, there is also many times the involvement of calcium, which is attributed, just like in the case of G-alpha Q. But overall, this is what is to be understood, that the CAMP, once it is formed through the activation of adenylyl cyclase from ATP, the CAMP will go and activate the protein kinase A, which will show the intrinsic activity. So this whole cycle deals with the G-alpha S subtype of GPCR.